If you dive deep into the ocean, you may encounter sharks that look far more terrifying and distorted than you would expect. At just around 1,000 meters below the surface, pressure increases to over 100 times that at sea level, comparable to your entire body being compressed by forces far exceeding the air pressure inside a typical car tire, which is only about 30 to 35 pounds per square inch. Under these conditions, the familiar form of sharks begins to change. Their bodies distort, their heads extend into specialized sensory structures, and teeth appear in positions that seem almost unnatural. This is not a rare exception, but a consistent pattern observed across increasing depths. But why would evolution effectively redesign one of the ocean's most efficient predators in such an unfamiliar way? Near the surface, the sharks follow a design that appears both familiar and efficient. The Caribbean reef shark is a clear example. It reaches over 3 meters in length, moves with streamlined precision, and relies heavily on vision and speed to hunt. Its body is balanced for hydrodynamics, allowing it to chase and capture prey in well-lit waters. There is nothing excessive or distorted in its form. Every feature serves a direct purpose. This is what a default shark looks like under stable environmental conditions. Abundant light, consistent food sources, and moderate pressure allow evolution to favour efficiency over specialisation. At this level, survival does not require extreme adaptation. Evolution does not aim to create beauty, symmetry, or even familiarity. It operates under a single constraint: survival within a specific environment. In biological terms, morphology is shaped by selective pressure. When conditions remain stable, organisms tend to retain efficient and generalized forms. However, as environmental variables shift, light availability, pressure, temperature, and food density, those same forms become insufficient. Adaptation then drives structural change, sometimes in ways that appear extreme or counterintuitive. A feature that seems excessive or misplaced often reflects a precise functional response to a constraint. In the case of sharks, their transformation with depth is not random distortion. It is a progressive reconfiguration where each modification corresponds to a specific limitation imposed by the environment. The first signs of deviation appear not in complete darkness, but just a few hundred meters below the surface. At around 200 meters, another basking shark introduces a form that already challenges expectations. It can grow up to 12 meters in length, yet its most defining feature is its mouth, which opens wide enough to filter massive volumes of water. Instead of hunting large prey, it consumes microscopic plankton. This apparent mismatch between size and diet follows a clear biological principle. Plankton is abundant but highly diluted, requiring continuous intake in large quantities. To sustain itself, the basking shark processes over 2,000 litres of water during feeding, using specialised gill structures to trap nutrients efficiently. In the twilight zone, where light rapidly diminishes, the saw shark presents one of the most unusual structural adaptations. Its elongated snout is lined with sharp, tooth-like projections, forming a rigid, blade-like extension. At first glance, this appears inefficient compared to a standard jaw. However, embedded within this structure are dense arrays of electroreceptors, allowing the shark to detect faint bioelectric signals emitted by prey hidden beneath sediment. In an environment where vision becomes unreliable, this sensory system replaces sight. Once a target is located, the saw shark swings its snout laterally, striking or trapping prey before using its actual jaws to consume it. The structure is not decorative or excessive. It is a precise solution to hunting in near darkness. Deeper within the twilight zone, the megamouth shark presents a different kind of anomaly. Unlike the basking shark, which is proportionally massive, the megamouth reaches only about 4 to 5 meters in length, yet its mouth can exceed 1.3 meters in width. And this disproportion creates a form that appears structurally exaggerated. Like other filter feeders, it consumes small organisms such as krill, drawing in large volumes of water and filtering nutrients through its gills. However, due to its rarity, fewer than 120 confirmed sightings, its behaviour remains only partially understood. One hypothesis suggests that the inner lining of its mouth may reflect or emit light, potentially attracting prey in low light conditions. This mechanism has not been definitively confirmed, but it illustrates how limited observation constrains scientific certainty. At this depth, it becomes necessary to distinguish between what is firmly established and what remains uncertain. The anatomical structure of the megamouth shark, its feeding method, and its reliance on filtering small organisms such as krill are well documented. These observations are based on physical specimens and recorded encounters. However, behavioural interpretations are far less certain. The idea that its mouth may emit or reflect light to attract prey remains a hypothesis, not a confirmed mechanism. This distinction is critical. In deep-sea biology, limited observation often leads to inferred explanations rather than direct evidence. As a result, scientific understanding progresses unevenly. Some features are precisely measured, while others remain speculative, constrained by the difficulty of studying organisms in environments that are rarely accessible to direct observation. In the midnight zone, where light no longer penetrates, and the goblin shark represents a shift from sensory adaptation to mechanical efficiency. Its most distinctive feature is its protrusible jaw, which can extend rapidly forward to capture prey. Unlike surface sharks that rely on speed and pursuit, the goblin shark moves slowly, conserving energy in an environment where food is scarce and unpredictable. When prey comes within range, the jaw launches outward with precision, reducing the need for prolonged chases. This mechanism is supported by specialized ligaments and cartilage that allow rapid extension and retraction. Its elongated snout, filled with electroreceptors, further enhances its ability to detect prey in complete darkness. The design prioritizes minimal energy expenditure while maximizing capture success in a low-resource environment. The frilled shark presents a structure that diverges even further from conventional shark morphology. Its elongated, eel-like body allows it to move with minimal disturbance in deep water, while its mouth contains approximately 300 teeth arranged in multiple rows. Each tooth is divided into 3 needle-like cusps, forming a backward curving trap. This design is specifically suited for capturing soft-bodied prey such as squid, which are both fast and highly slippery. Instead of relying on forceful bites, the frilled shark uses a sudden forward lunge, similar to a snake strike, to intercept its target. Once contact is made, escape becomes extremely difficult. The more the prey struggles, the deeper it becomes entangled in the hooked teeth. This is not a system built for cutting. It is optimized for retention under conditions where missed opportunities are costly. The Greenland shark represents one of the most extreme biological adaptations known among vertebrates. It inhabits cold, deep waters of the North Atlantic, where temperatures remain near freezing and metabolic processes slow significantly. Unlike most sharks, it moves at a speed of roughly 1 to 2 kilometers per hour, conserving energy over long periods. Its most remarkable characteristic is its lifespan. Radiocarbon dating of proteins in its eye lenses has revealed individuals living at least 272 years, with some estimates approaching 400 years. Growth is extremely slow, and sexual maturity may not occur until around 150 years of age. In many cases, parasitic organisms attach to the eyes, impairing vision, yet the shark continues to function effectively by relying on olfaction and low-frequency sensory cues in near-total darkness. At extreme depths, biological adaptation reaches a fundamental limit. As pressure increases, it begins to disrupt the structure of proteins, which are essential for cellular function. To counter this, deep-sea organisms produce trimethylamine N-oxide, or TMAO, a compound that stabilizes proteins under high pressure. However, this solution is not unlimited. As depth increases, the required concentration of TMAO also rises. Beyond a certain threshold, it interferes with normal biochemical processes, effectively becoming toxic to the organism itself. Sharks, in particular, already maintain relatively high baseline levels of TMAO compared to other marine animals. This creates a constraint, because while they can tolerate significant pressure, they cannot indefinitely increase TMAO without compromising internal function. Combined with their relatively rigid bodies, this sets a hard boundary on how deep sharks can exist. Despite these well-defined biological limits, several observations do not fully align with expected distribution patterns. Many deep-sea sharks have not been discovered through systematic exploration, but rather through accidental encounters. The first recorded megamouth shark, for example, was found in 1976 after becoming entangled in a US Navy sea anchor at a depth of approximately 165 meters. Similarly, goblin sharks, typically associated with depths exceeding 1,000 meters, have occasionally been captured much closer to the surface. And cookie-cutter sharks have been documented leaving circular bite marks not only on marine animals, but also on submarine cables and equipment. These events are rare but consistent. They suggest that the vertical movement of some deep-sea species may be more dynamic than previously assumed, or that external factors influencing their distribution remain insufficiently understood. These observations lead to a more precise question. If depth alone defines the structure and behavior of these sharks, why do some individuals appear outside their expected environmental range? Vertical migration is a known phenomenon in marine biology, particularly among smaller organisms that follow daily light cycles. However, for large deep-sea sharks, such movement is less understood and rarely observed directly. Could fluctuations in temperature, pressure gradients, or prey distribution be sufficient to explain these anomalies? Or is there an additional environmental variable that has not yet been fully measured, such as localized electrical fields or chemical gradients? The current data does not support a definitive conclusion. What can be stated with confidence is that the deep ocean remains an incomplete system where observed behavior occasionally exceeds existing explanatory models. Long before deep-sea exploration became possible, many of these organisms were interpreted through incomplete observation. Sailors in the 18th and 19th centuries reported encounters with serpent-like sharks, or creatures with unnatural jaws and elongated heads. Without the ability to observe them in their natural environment, these descriptions were often exaggerated or misclassified. Circular wounds found on whales and large fish were once attributed to unknown predators or even non-biological causes. It was not until the identification of the cookie-cutter shark that these marks were accurately explained. These historical accounts were not fabricated, but they were filtered through limited knowledge. What appeared anomalous or unnatural was, in many cases, a direct consequence of biological specialization under extreme environmental conditions. Human perception is not neutral when confronted with incomplete information. In low visibility or unfamiliar environments, the brain tends to prioritize threat detection over accuracy. This is a well-documented cognitive bias. When an organism appears structurally unusual or behaves in ways that are not immediately understood, it is often interpreted as dangerous or unnatural. In the context of deep-sea sharks, features such as protruding jaws, elongated snouts, or abnormal dentition can trigger this response. Without direct knowledge of their function, these traits are easily misclassified as anomalies rather than adaptations. Over time, repeated misinterpretations reinforce collective assumptions. This process explains why deep-sea organisms are frequently associated with fear or labelled as monstrous, despite operating within consistent biological constraints. The strongest conclusion is also the most limited one. Deep-sea sharks are not evidence of anything supernatural, nor are they arbitrary biological distortions. Their unusual forms are best explained as adaptive responses to darkness, pressure, scarce food, and the physical constraints of life at depth. At the same time, not every feature or movement pattern has been fully resolved. Some behaviours remain insufficiently observed, and some hypotheses remain untested because the environment itself is difficult to access. That uncertainty matters. It does not weaken the science, it defines its boundary. What appears mysterious is often a sign not of hidden forces, but of incomplete observation in one of the least accessible regions on Earth. What emerges from this progression is not a descent into chaos, but a gradual narrowing of what is biologically possible. Sharks do not become distorted without reason. Each structural change reflects a constraint: less light, less energy, more pressure, fewer opportunities. As these constraints accumulate, the familiar form breaks down into something that appears unfamiliar, even unsettling. Yet the underlying logic remains consistent. These organisms are not anomalies, they are solutions. At the same time, the deep ocean remains only partially observed. Large regions have never been directly explored, and many species have been documented only once or twice. This leaves open the possibility that additional forms exist beyond current records, not as unknown forces or unexplained phenomena, but as extensions of the same adaptive processes already understood. The deeper we go, the less the ocean reflects our expectations. And perhaps that is the central point. These environments are not designed to be understood intuitively. They follow rules that are precise, but not always visible.